Pooja is facing some network issue.
Okay,
I
okay.
think. Yeah.
Should we wait for a couple of minutes
No,
or should
no,
I?
sir. No, sir. You can start.
Okay.
Yeah.
Okay. Hello.
Yes, sir.
Yes, sir.
Just a second. I'm trying to get my presenter view. Okay. Can you see my pointer here to the left here?
Yes, sir.
Yes, sir.
Top,
Yes, sir.
Yes.
right,
Yes, sir.
Yes, sir.
and bottom.
Yes,
Okay.
sir.
Yes.
So last time we were talking about protein synthesis, and we had just finished the three main steps to get the protein, which is initiation, elongation, and the termination and release of the protein. And we had just started on looking at what happens after this protein is released. Okay, and we said that in many cases, okay, what you find is that after the protein is made, it is further modified, and this modification after the process of termination is referred to as post-translational modification. Okay? and uh, and this modification occurs both in prokaryotes as well as eukaryotes, but to a much greater extent in eukaryotes. Okay, and some of the modifications that we listed were as follows: amino terminal modification, carboxy modifications, what do you call it, loss of signal sequence, etc., etc. And we started looking at each one of them separately. Okay, so the first type of modification was amino terminal modification and carboxy terminal modification. Okay. Can you just check if anybody's mic is on? Okay. Is that better? Hello? Hello?
Sir, sorry, so it
Yes,
was
sir.
mine.
Okay, okay, okay. So, so if you look at the first type, which is amino terminal modification and carboxy terminal modification, these are exemplified by the following. Okay, now remember that in prokaryotes, when protein synthesis begins, the first amino acid added is always formyl methionine. While in eukaryotes, it is a regular methionine. But what you find in many cases is that in the final protein, you don't find this formyl methionine or methionine, which means that something happens to the end of the protein after translation where these first amino acids are removed, or sometimes even more than one amino acid is removed. Okay. Usually what one finds is that generally more than one amino acid is removed, and then in addition to that, many times the N terminal is acetylated. Okay, what you form N acetyl amino terminal. Okay, and the reason for this is again manifold, but what is important at this point is to realize that the many proteins in our body do not have a methionine at the N terminal or formyl methionine in case of bacteria, simply because after translation it is removed. Okay. Likewise, you also find changes on the C terminal amino acid. You will find that sometimes C terminal amino acids are removed, or they are also modified by esterification. Okay. And this is, I think we started looking at it last time, where we said if an N terminal, nothing has happened, then the N terminal would look something like this, and it would invariably be, as I say, a methionine or formal methionine. Okay, but the modifications that you see many times is that you remove many of the amino acids from the end, and then you might add a formyl group or add an acetyl group, or sometimes even a propionyl group. Now, these are not the only modifications that are seen. Sometimes you also see what is called as monomethylation or dimethylation or trimethylation with a charge at the end terminal. Okay, or you might find something like this, where you have S palmitylation, especially the first amino acid is a cysteine, where to the sulfur is a thioester form, or even the amino terminal can be modified by amide formation with a fatty acid. Okay, or if you have a amino terminal glutamate, it can kind of ring close to give you a cyclic structure. So very unusual changes you can see at the N terminal. Likewise, the C terminal, as I said, can be esterified, form a methyl ester, or it can be converted to an amide. In addition to this, you also find that many times a stretch of amino acids from the N terminal is removed. Okay, and the reason for removal is that the function of some of the amino acids at the N terminal is to actually direct the synthesized protein to different parts of the cell. Okay, and this is shown here. For example, translocation into the endoplasmic reticulum directed by amino terminal sequences of some eukaryotic proteins. So here are some eukaryotic proteins where the amino terminal, which is shown here in kind of yellow, is responsible for directing them to the endoplasmic reticulum. And once the protein reaches the endoplasmic reticulum, then these sequences which are at the end or the end terminal are all removed by cleavage, and that is shown here with a red arrow. So in the end, you'll find, for example, human influenza virus A, it makes a protein which is this big, then this is directing the protein to the endoplasmic reticulum, and once the protein gets there, this entire piece is removed. Okay, so this is a very common modification that you see, where you lose an entire stretch of amino acids from the end. Another is that after the protein is made, several of the side chains of the amino acids which are part of the protein can also be modified. Okay, and here are some very common examples. Okay, you have already heard of this phenomena, where you have phosphorylation and dephosphorylation, which allows a protein to be many times active or inactive. And these phosphoryl groups are added to serine side chains, or threonine side chains, or sometimes tyrosine side chains, which are present in the protein. So the protein can toggle between what is called as phosphorylated and dephosphorylated stage. And this reaction is done by what are famously referred to as protein kinases. You must have heard of this. Protein kinases are enzymes which put a phosphate group on a protein, and this phosphate group is derived from ATP. Okay, so this is a product that you get after protein kinase works. The amino acid side chain is phosphorylated. Okay, here the reaction is shown with the amino acid, but actually speaking, you have to look at it as a big protein where there is a side chain serine and that is phosphorylated. In this case, also a big protein side chain is tyrosine and that is phosphorylated. Another interesting modification that you see on the side chain of a amino acid is that in one of the clotting factors, which is responsible for our blood being able to clot, one of the side chain glutamates, which is normally CH2, CH2, and COOH, is modified, and is modified by addition of another carboxyl group, okay, which is called as gamma carboxylation. So you end up getting a gamma carboxyglutamate, and this modification is very, very important for the functionality of the protein in the whole clotting process. So if you inhibit this process, then you affect the clotting process and your blood doesn't clot correctly. Okay? Another type of modifications is, as I said, we already seen this before at the end terminal, but this can also happen in the middle of the protein. Like if you have a lysine, it can get methylated or dimethylated or trimethylated. Likewise, glutamates in the middle of the protein can get esterified to give methyl glutamate. Okay, so these are all modifications that are occurring on the amino acids which are present as part of the protein after the process of translation. Even more interesting is what is called as glycosylation, okay, where sugars are added onto the side chains of proteins. And typically, what you find is you will have sugars attached to serines or what do you call it, asparagines, and so on and so forth. Okay, here is an example. This is a side chain asparagine. Okay, aspartic acid normally is CH2COOH, and when it is an amide form, it is CH2CONH2. Now, to this NH is attached the sugar. Okay, in this case, it is N-acetyl glucosamine. So, N-acetyl glucosamine is added on to the asparagine, which is part of the protein. Okay, now sometimes this glycosylation patterns can be very complex and interesting. Here is an example where on an asparagine or a serine are added a series of sugars. Okay, and here is shown what blue means. Blue means N-acetyl glucosamine. Green means mannose. Yellow is galactose. Yellow square is N-acetyl galactosamine. Red, diamond shape is neuraminic acid, 5-acetyl neuraminic acid. Triangle is fucose. Okay, so you can have very interesting patterns, and this is as an offshoot from the main chain of the protein. Okay, and many times you'll find a lot of proteins which are part of the membrane on the surface will have the sugars attached on the protein, and the sugars play a very important role in recognition processes. Okay, just for example, some of the sugars are also the ones which determine which blood group you are, A, B, AB, O, etc., etc. Okay, so one of the ways your blood is grouped is based on some of the sugars which are present on the proteins on the surface of some of your cells which are present in the blood. Okay, and even more complex are C patterns, and this is not restricted to only humans. Huh? You can find this thing in plants, in insects, in animals, and yeast. A whole bunch of sugars. They all get attached to the protein, either onto asparagine or a serine or a threonine. Okay, now when it is attached to an asparagine, the linkage is always via nitrogen. So they are called as N-linked sugars. But when it is serine or threonine, it is always through oxygen. So they are called as O-linked sugars or O-linked glycans. Okay, and you can have very complex patterns. Okay, and what I'm showing you is some of the simpler ones, but they can be very elegant and very different. And therefore, because of this pattern, you can have different recognition of the surface of the cells by different ligands. Okay. Another modification is what we call as isoprenylation. Okay, and remember, you have seen isoprene units. They are the ones which come together to form, cholesterol, etc., etc. And see what happens in this case. Okay, uh, 2 or 3 of these 5-carbon units come together to form geranyl phosphate or farnesyl phosphate, and this then gets attached onto the sulfidyl group of a protein side chain cysteine. Okay, so this S of the side chain cysteine takes up this long chain, and you get what is called as farnesylated protein. Okay, you can get geranylated proteins, farnesylated protein. Sometimes entities like ubiquitin also bind. You get ubiquitinylation, as they call it. So all these are modifications where you have got a long chain of hydrophobic carbons attached to the protein. Okay, and they also play a role in signaling and directing the proteins. Okay. The other modification is addition of prosthetic groups. Okay, you know already by now that a lot of enzymes for their function need either cofactors or prosthetic groups, where these groups are part and parcel of the functional enzyme. It's like they're always there. Okay, for example, in vitamin B12, you need this porphyrin ring for it to be active. Okay, and this is shown here as iron here, that is cytochrome P450. See, this cobalamin is here. See, this cobalt is in the middle. So all these things are not actually proteinaceous in nature. They are added later on after the protein is made. Okay. Similarly, you'll have many proteins where you need to have biotin attached via lysine as a prosthetic group. Remember when we studied vitamins? I don't know whether I taught you vitamins or not, but one of the vitamins is biotin, and biotin is a vitamin which is plays a role in carboxylation of proteins. And this is biotin is always present as part of the protein attached to the lysine. So you see the side chain CH2, 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 and that is the lysine of a protein. To that is attached biotin, and this is biotin. And biotin is a prosthetic group. Okay, this is a prosthetic group which is present in P450. This is cytochrome, what is called porphyrin ring with iron in the middle. This is also similar to a porphyrin ring, but it has got a cobalt in the middle, and that's why entities which contain this are called as cobalamines. Okay, so these are all added after the protein is made. Okay, and proteins which use cobalamine as a cofactor, or cytochrome, or what you call as porphyrin ring as a cofactor, they have these things attached to them so that they can be functional. The other modification is what you call as cutting of the big protein, or modification of big protein by what you call as proteolytic processing, as it is called. Okay, here is an example of that. Okay, this is chymotrypsinogen. It is produced as a big 245 amino acid protein. Once it is trypsin. Once it is produced, trypsin acts upon it and it removes the first 5 amino acids. Okay, and then it leaves the remaining 16 to 45. So you get what is called as chymotrypsin. Okay, and this chymotrypsin again is further broken down, where the terminal part is removed, 149 to 249, and you end up getting 3 chains now: A chain, B chain, and C chain. Okay, and the activity is then based on removing of bits. Okay, so this is inactive as a trypsinogen, but this is active. Okay, now look at the other example. Here is a trypsinogen. Okay, there's an enteropeptidase which breaks trypsinogen to give you trypsin. Okay, what does trypsin do? It takes pro-carboxypeptidase, cuts it to give you carboxypeptidase. It takes pro-elastase, cuts it to give you elastase. It takes chymotrypsinogen and breaks it to give you chymotrypsin. Okay, it can also take pro-lipase and break it to lipase. It's very interesting. This is a protein which activates all of these fellows by taking out a piece of, a sequence of peptides. But it itself is activated by another peptidase which takes a piece out of trypsinogen to give you trypsin. Okay, so here in all of these, if you look very interestingly, there is a proteolytic processing. That means a peptide removal based activation of a protein. Okay. Another interesting post-translation modification is disulfide bond formation. Okay, and disulfide bond formation is very, very common in many proteins, and you can get what are called as intra-strand disulfide bond formations and inter-strand disulfide bond formation. Okay, here is an example of insulin. Insulin, when it is produced as a pre-proinsulin. Okay, and once it reaches the vesicles where it is stored, this signal peptide or the signal sequence is removed to give you this shorter protein, which is called as proinsulin. If you look at proinsulin, here is one disulfide bond which is formed, and this is what is referred to as intra-strand disulfide bond. There is another disulfide bond. There is a third disulfide bond. Now, once these disulfide bonds form, the protein is, in a sense, put into the correct shape. And once it is in the correct shape now, then this pink region here, which is called as a connecting peptide, is also cut out, and you end up getting functional insulin, where you have the A chain and the B chain, and are correctly set, and it is only when it is present in this form that it is active. You also see disulfide bonds in antibodies. In antibodies, there is an example where you have a disulfide bond between two heavy chains. Okay, these are the heavy chains, if you remember, and these are called as the light chains. Now, heavy chains and light chains are also attached to each other or interact with each other to disulfide bonds, which are called as interchain disulfide bonds. But there are also disulfide bonds within the light chain and within the heavy chain. These are called as intra-strand disulfide bonds. So all these bonds are very important so that the protein attains the correct three-dimensional shape for it to be functional. Okay, and if there is any problem with this disulfide bond formation, then you also have issues in terms of activity of the protein. Now, if you remember in biotech, we talked about the fact that when they manufactured insulin, they produce A chain separately and they produce B chain separately. Now, when they mix it together, when the two chains are separate entities, what happens is this disulfide bond which is formed sometimes doesn't form correctly. For example, this sulfide forms a bond with this one, then this forms a bond with this one, and so on and so forth. And when that happens, you end up getting A chain and B chain linked to each other, but it doesn't show any sugar lowering activity because it is not correctly structured. Okay, and only when the disulfide bonds are correctly formed will the insulin be active. And remember, I told you that that's why when they first made insulin by recombinant DNA technology, the yields of active insulin were very, very low. So in the next approach, which was started like about 5 years after they made recombinant insulin, they actually made the sequence for the whole pro-insulin and inserted that into the bacteria. So when the bacteria produced this big chain, then this disulfide bond formation was helped because all the amino acids are in the correct position. And then what they did was synthetically remove this chain by treating with trypsin so that the yields of active insulin were much, much higher. Okay. In addition to this, we are also familiar with the fact that proteins have quaternary structures. There are a lot of proteins which are active only and only when they interact with other proteins. Okay. And sometimes these interactions may be between what are called as like subunits. Okay. So you might have what is called as multimers, which are composed of the same monomer. Okay. For example, this is collagen, and collagen is composed of three peptide chains which are identical, but they are interacting with each other through what is called as a helix. Okay, formation. So this is a triple helix, as it is called. Okay. Likewise, you have hemoglobin, which is made up of two different types of proteins, which are referred to as A chains and B chains, and two A chains and two B chains come together to form what is called as a functional hemoglobin, which has got a very high capacity to carry both oxygen as well as carbon dioxide. Okay, so and which means that once I make the A chain, I cannot expect activity from it unless, of course, it interacts with another A chain and a B chain. Okay, so all of these modifications are all necessary in order for the protein to be functional, and all these are said to be post-translational modifications. And as I said, uh, these occur to a much greater extent in eukaryotic systems and to a smaller extent in prokaryotic systems. Okay, but you need to get a feel for this, and many times the way this post-translational modifications are done allows you to even regulate the activity of the protein. For example, I can store a protein as chymotrypsinogen for a long time and just cut it out when I need the activity. Or I can store something as pre-pro-insulin for a long time and cut it out when I reach the vesicle and store it as pro-insulin, and then again cut to give me active insulin. Okay, so it gives you a level of regulatory capacity when you have these PTMs which convert protein from inactive to active or active to inactive. Okay, now because protein synthesis is so crucial to the survival of an organism, you'll find that many drugs which are used for treating infections mainly are protein synthesis inhibitors. Okay, and by inhibiting protein synthesis very specifically of say the pathogen, you are able to then in a sense inhibit the growth of that pathogen and treat the infection which is caused by the pathogen. Okay, let us look at some of the compounds which interfere with the protein synthesis process. Just like we looked at some compounds which interfere with DNA replication. If you remember, things like thiopurine and intercalating agents and alkylating agents, etc. In the same way, there are some drugs which are used which are based on their ability to inhibit protein synthesis. Okay, so here is the first one. It's a drug which is called as puromycin. Okay, so look at puromycin. This is the structure of puromycin. Okay, and if you think look very closely, it actually resembles the end of the tRNA. If you remember, the end of the tRNA has got an amino acid attached to it through a ester bond, which we call as the activated tRNA. Okay, so this puromycin has got resemblance to the end of the tRNA, and because of that, what happens is it is able to come and sit in the ribosome, and what it does is that it sits in the ribosome as if it's a tRNA, and then when the protein synthesis occurs, it uses its amino group, which is present here, to form a amide bond with a peptide which is already there as part of the protein synthesis process. And what it basically does is, whatever is the peptide which is synthesized, it attaches to itself. But the problem is, it itself is not amenable to further reactions. So what happens is, after that reaction occurs, the peptide gets transferred to puromycin, and puromycin simply leaves, basically giving you what is called as truncated protein synthesis or interrupted protein synthesis. Sometimes you end up getting dipeptides, tripeptides, pentapeptides, depending on the concentration of puromycin. Essentially, what it does is it inhibits synthesis of complete proteins by basically prematurely terminating by taking the peptide which is formed and doing a reaction on itself because it has got affinity for this A site, because it resembles, in a sense, tRNA. Okay. Then we have tetracycline and chloramphenicol. We have heard of these two antibiotics. What tetracycline does is blocks the A site of the bacterial 70S ribosome. Okay, so this is the A site and this is the P site. So what does tetracycline do? It sits in the A site. And what does chloramphenicol do? It affects the activity where the peptide bond is formed. It affects what is called peptidyl transferase activity of the 70S. And this specifically happens to a greater extent with respect to the bacterial systems, and therefore they are used as antibacterial. Okay, so as I said, this peptide bond formation is inhibited by chloramphenicol, and tetracycline just sits here and it affects the protein synthesis by binding to the A site. There is another cycloheximide. Okay, but look at cycloheximide. It blocks peptidyl transferase activity of the ATS ribosome. That means what? It is selective for eukaryotes. Okay, it doesn't have too much activity against prokaryotic systems. It is very specific for eukaryotes. So if you give a human cyclohexamide, then you can affect the protein synthesis of the human, and therefore, in that sense, it will be toxic to humans. Okay, streptomycin, which is also another antibacterial, causes misreading of the mRNA. Okay, so when it is present, then the ribosome tends to slip, and when it misreads the mRNA, you tend to form sometimes faulty proteins, or you read something as a stop codon, and the synthesis stops. Okay, so these things can happen there. Okay. Uh, then another very interesting compound which affects protein synthesis is diphtheria toxin. Okay, produced by Corynebacterium diphtheria. Okay, and see what it does. Okay, it causes what is called as ADP ribosylation. Okay, ADP ribosylation is where ADP ribose. ADP ribose means adenine plus sugar, which is adenosine, and ribose with this, and two phosphates attached to the ribose. So that's why this is called as ADP, adenosine diphosphoRibose. Okay. And this ADP is attached to diphthemide, and diphthemide is a part of See this eukaryotic elongation factor. Okay, so this elongation factor has a histidine as a side chain, and this elongation factor has histidine which is modified. So see the modification. Okay, so here is a histidine. This is a side chain of histidine. Remember, imidazole amino acid. Now to that carbon which is present in between the two nitrogens is attached this amino acid. Okay, this amino acid is kind of like, if you can think, no, a variant of alanine. Okay, NH2 CH CH3 is alanine. And remember, if I add another carbon to alanine, I'll have extra carbon, and that extra carbon comes from S-adenosyl methionine. Okay, and you get this link. Okay, the other way to look at it is the side chain, which is CH2 and then SCH3 is also the side chain of methionine. Okay, so even that can be used in order to produce this side chain. Now see what happens after this side chain is produced. After this side chain is produced, to the nitrogen is added a lot of methyl groups. Okay, see this methyl groups, CH3, and it's converted to a what do you call as quaternary ammonium compound. Once this reaction occurs on elongation factor 2, then this elongation factor is able to do all its function. But see what diphtheria toxin does. Diphtheria toxin takes this diphthemide portion of the elongation factor, and to it, it attaches what is called as a ADP ribose. And where does it attach the ATP ribose? To this nitrogen. And when this is attached to this diphthemide, then this elongation factor is non-functional. It cannot do its job, and therefore protein synthesis comes to a standstill. And this happens to eukaryotic cells. That's why when you get infected by Corynebacterium diphtheria, which causes what is called whooping cough, one of the ways the bacteria is able to cause problems to your cells is to inhibit the synthesis of proteins inside your cells, and that's where you see all the effects of the infection. Okay. Another very interesting toxin is ricin, and ricin is present in Ricinus communis. If you remember, if you are familiar with ricin, anyone know what
Castor oil.
Castor, yeah.
Yes.
And castor oil, if it is not treated correctly, if you consume castor beans raw or castor oil without treating, then it can actually kill you. It can kill you because of the presence of this ricin and abrin, which are both compounds which affect eukaryotic protein synthesis. In fact, if you look at all the toxins which are available in this world, both diphtheria toxin as well as ricin, they make it to the list, okay, of being one of the most toxic compounds. Okay, just to give you an idea, there are some of the other toxins. The most toxic compound is called botulinum toxin. Okay, then you have dioxin, which is also very toxic. A cyanide is somewhere here, LD50 or 4 milligram per kilogram. Okay, and then just to give you a feel, anything which you think is safe is also toxic if you take too much. For example, if I take salt to the extent of 3,320 milligrams per kilogram, that is what it basically means, 3 grams per kilogram. So if I give you, if you're a 70 kg person, and I give you 250 grams of salt and tell you to swallow it immediately, I can kill you with that. In fact, I can even kill you with water. If I give you 90,000 liters of water and tell you to drink it, I shouldn't say 90,000 liters, 90 grams of water per kilogram. Okay, assuming a density of 1, and I tell you to drink that much water in one go, I can kill you. Okay. Just as a little side thing, the LD50 of abrin and ricin are given here in nanogram per kilogram. Okay, so these are fairly toxic compounds. And in this here, if you look, there is a diphtheria toxin, okay, which is in a sense very close to botulinum toxin. Okay, so very toxic compounds, and they all work by inhibiting protein synthesis. Okay, so so just to summarize very quickly, in prokaryotes, if you remember, transcription and translation can occur at one time. Here, transcription always occurs in the cytoplasm, and then translation occurs in the transcription occurs in the nucleus, and translation occurs in the cytoplasm. Here, the polyribosomes, as I say, are linear, and here they call it circular. Okay, uh, and that is because of the arrangement you get when you are actively making proteins with many ribosomes at one time. No capping of mRNA, capping of mRNA. Start codon is AUG next to the ribosome binding site, which is the Shine Dalgarno sequence, or nothing like that. Okay, so the first AUG here is used as a start codon. First amino acid always formal methionine. First is methionine, but not modified. 70S ribosome, 80S ribosome. Then all the other things. You have elongation factors and initiation factors, limited number. Here there are many more. Okay. And as I say, you can shut off protein synthesis by dimerization of ribosomes in non-growing cells. We have not talked about this. Here, we do it by controlling the extent of elongation. Controlling the availability of initiation factors. So that's how we control protein synthesis. Okay. Now, I also want to go ahead and just get this thing started. We have looked at how nature makes proteins. Okay, it does so by looking at mRNA to direct the sequence of proteins that need to be put together, and then it uses the ribosomes and tRNA, which are carriers of the correct amino acid, to direct the protein synthesis so that it occurs in a very systematic fashion from the N terminal to the C terminal. Just like nature makes peptides or proteins, one can also make peptides or proteins in a laboratory. Okay, basically, you need to form a peptide bond between the amino group of one amino acid and the carboxy group of another amino acid, and you should be able to form a peptide. Okay, just to show here as an example, let us say this is one amino acid, this is another amino acid, and this is a third amino acid where the R groups are different. If I have a way of very systematically forming a amide bond between this amino group and this carboxy group, and this amino group and this carboxy group, then I'm able to connect all the three amino acids in a very specific sequence, and then I'll get a tripeptide with a given amino acid as an N-terminal amino acid, which has the R group. The R double prime group containing amino acid ends up forming the C terminal, and this amino acid, which has got R dash side chain, forms the middle amino acid of the tripeptide. Okay, all said and done. But when I try to do this in a laboratory, there are a lot of problems which arise. Okay, and the problems arise because all amino acids have two functional groups. Okay, so if I'm trying to take a mixture of glycine and alanine, and in my mind I want the amino group of alanine to form a peptide bond with the carboxy group of glycine to give me this dipeptide. The one I want is NCH2CO NH CH3COO. So I want this dipeptide which will be called as glycyl alanine, which is shown here. When I try to do this in the laboratory in a test tube, because of the presence of multiple functional groups, I have a lot of difficulty in preventing the formation of the other three. Look at what happens in this case. Here, the amino group of alanine attaches to a carboxy group of another alanine to end up giving me alanine. Likewise, the amino group of glycine attaches itself to the carboxy group of another glycine, and I end up getting glycine as a dipeptide. Or instead of this amino group reacting with this carbonyl carbon, this amino group ends up reacting with this carbonyl carbon, and I end up getting alanyl glycine rather than glycyl alanine. Okay, and this is the problem which we face when you're trying to make a dipeptide. So you want one dipeptide, but you end up getting a mixture of four, so your yields become only at the most 25%. Okay, if you are lucky. Okay, Now, is there a way I can direct this bond formation so that only one type of reaction occurs and I get the dipeptide of my choice? Okay. So the way one does it is this. Okay. So what is done typically is you take one of the amino acids which you want to be the N-terminal amino acid and you block the amino group of that one. Okay, so see what I've done now. I will take this glycine and I'll somehow cover this amino group so that it becomes an unreactive. At the same time, what I do is I take this carboxyl group and I activate it. And I activate it so that it will have a higher affinity for a nucleophile so that the reaction tends to occur at this carbonyl carbon. Okay. At the same time now, see what I do? I take this second amino acid and I do not do anything to the amino group. I keep it open, I keep it active. And then if I want to be extra sure, I will again protect this carboxyl group so that this cannot react. So by doing that, what I have done is made sure that this amino group can only react with this carboxyl group and I'll only get one type of dipeptide. Okay. So now what will happen? If see how I do it, I will take glycine for example, and I'll treat it with what do you call it? Di-tertiary butyl dicarbonate. Di-tertiary butyl. That is di-tertiary butyl, and this is dicarbonate. When I do that, this amino group of glycine reacts with this carbonyl carbon, and it picks up this tertiary butoxy carbonyl group and it gets protected. So now the amino group will not be able to react because it is capped with this tertiary butoxy carbonyl. And these are the other byproducts of the reaction. Okay. The other way I can protect the amino group is by treating with what is called as either tertiary butoxy carbonyl or what is called FMOR group, where you have fluoridine methoxy. Okay, so so this is not the only way to protect the amino group. You can do so by, as I say, treatment with BOC or FMOC. Now see what I do. I have this protected amino acid. I react this carboxyl end with a reagent called as dicyclohexyl carbodiamide. Dicyclohexyl carbodiamide has a very electropositive carbon, okay, or what you call electron deficient carbon because of the presence of two nitrogens adjacent to it, which are electronegative, which pull the electrons. So what happens is when I adjust the pH to be slightly basic, this carboxylic acid group will ionize. The CO minus will attack this carbon and will transfer this bond up here, which will pick up a proton. And by doing so, essentially what I do is I activate this carboxyl group. Okay. So now I have protected the amino group and I have activated the carboxyl group. Now all I have to do is introduce the second amino acid, and when the second amino acid comes in with its lone pair of electrons, it has a very high tendency to react with this carbonyl, because when it reacts with this carbonyl, it can eliminate this group as a good leaving group. And when this leaves as a leaving group, you end up getting dicyclohexyl urea. At the same time, now we have bound the peptide bond. Okay. Now, because this carboxyl group is very reactive relative to this carboxyl group, you have minimal reaction between this amino acid to give you what are called as homomers. Okay. So the amino group of this amino acid will have a less tendency to react with its own carboxyl group on another molecule because it is not activated as this carboxyl group. So in a sense, this reaction will dominate and you'll end up getting the dipeptide of your choice. Okay. Now once I do this, now I have got a dipeptide. Now I'll take again this carboxyl group and react it with a fresh molecule of carbodiamide. And once I do that, this will get activated. And then again I'll add a third amino acid and so on and so forth. Okay. So now a dipeptide, I react it to DCC, I add the second, and I'll get the dipeptide, and so on and so forth. And I can keep on adding. Okay. So here the amino acids are adding to the growing C terminal end, just like it happens in your body. Okay, and when the desired number of amino acids are added very systematically, then in the last step, all you have to do is treat the final peptide with trifluoroacetic acid and methylene chloride. And what that does is it removes the protecting group here to give you the final amino acid, which is the one that you want. Okay, so this is what is called as laboratory peptide synthesis. However, this way of making peptides is nice, but it's now rarely done. We do peptide synthesis in the laboratory using what is called as solid phase peptide synthesis. Okay, and solid phase peptide synthesis is an improvement, as I say, on this liquid phase peptide synthesis, and many times it is referred to as Merrifield automated solid phase synthesis of a peptide. Okay, what this is, we will see next time. Okay, so I'm going to stop and let us see if we can go back to our thing. Let us see anything in the chat box. Okay. How is glycosylation? The first question is, how is glycosylation different from addition of a prosthetic group? Since the added sugar in glycosylation is also a prosthetic group. Okay. Okay. Now the difference is this: when let us say the sugar is part of the catalytic function of the enzyme, then that sugar could be called as a prosthetic group. Okay. But if you notice, in many proteins, sugars are added, but the proteins themselves are not enzymes, so they don't have any catalytic function. So when that happens, you do not use the word prosthetic group. The word prosthetic group is generally used when you have an entity which is added to a peptide or a protein which is an enzyme, and that this group then also helps in the catalysis reaction. Okay, so yeah, you are right in a sense that it is just like any other entity which is added to a protein. But the difference is when sugars are added, in many cases, they are on the surface of the protein and they only have recognition function. They do not have any role in any catalysis. If they did, then they would also be called as prosthetic groups. Okay. Here is another one. Let me see. We find proteins in different forms in the body. Some in alpha, some in beta, some in triple helices, some quaternary. So how do the protein take on these respective shapes? Okay. Is there some system in place for this purpose, and is there a relationship or a sequence to the type of structure? Yeah. In fact, if you were to think carefully, uh, every function of the protein or everything a protein can do, that ability is coded for in the sequence. Okay, so the correct sequence of amino acids is the bottom line and will determine finally and eventually how the protein will function. Okay, so in that regard, the what you call as the primary structure or the sequence of amino acids which make up a protein is probably the most important part of the relationship between the structure and the function. Okay, how do proteins take on these structures? Some proteins tend to fold what you call as on their own. Okay, they seem to follow this path of protein folding, which is thermodynamically most feasible and occurs without any assistance. In other cases, folding is helped by another group of proteins which are called as chaperones. Okay, so chaperones are proteins which help other proteins fold correctly and take on the correct shape. Okay, so there is some involvement of these chaperone proteins which also assist in a protein taking the correct shape. Okay, why are there alpha helices, beta plates? Alpha helices are determined by the nature of amino acids which are part of the sequence. Similarly, certain amino acids, when they are present in a row, what you call as make the peptide take up a beta sheet in preference to alpha helix. Okay, then how these alpha helixes and beta sheets come together in a tertiary structure is again sometimes determined by pure thermodynamics, or as they say, chaperone assisted taking up of that three-dimensional shape. Okay, and then proteins interacting with each other. That is also part of evolution to give you proteins which have quaternary structure. Okay, and remember, this is not all. Proteins can also interact with RNA. Proteins can interact with DNA. Proteins can interact with sugars. Proteins can interact with lipids. So you get lipoproteins, glycoproteins, nucleoproteins, and all of these come together to do their functions. Okay. Is there a system for place? Yeah, there is a system for place for doing all these things. As I said, there is a regulation at the level of chaperones, level at the regulation, the level of the sequence itself, which is the information that is carried by the organism. And again, the sequence does dictate what the function will be. Okay, does that answer your questions, Shivam and
Yes,
Aditi?
sir.
Okay. So let me do one thing very quickly. I'll download the attendance list. And again, any question, anyone? You can unmute and ask if you wish, or you can put it in the chat box like Shivam and Aditi do. Okay. If not, yeah.
No question, sir.
Okay. So can I stop recording?
Yes, sir.
Okay. So I'm going to stop the recording.